So hello everyone. I welcome you all for today's session. A very good morning to all of you. Am I clearly visible, audible to you people? Let me confirm it. First, let me confirm it in few seconds. If I'm clearly visible, audible, I will start the lecture ahead. Just a second. Give me a second. Okay. It will take few seconds to stream live. Okay. I hope it is working. Okay. So I welcome you all for today's session and I am Dr. Priyanka Sachdev here and I welcome you all. And today I am going to start this session with previous year questions of INICET. We know INICET is just about to come, it is ahead, and most of the students want me to take this session. So I have already taken previous year questions of INICET, both INICET, that is June 2021 and November 2020 of pathology. So today I am going to take this session for microbiology previous year question from INICET. So here I am. So before starting the lecture, let me tell you something, some announcement. So on Unacademy, we have two types of paid subscription. One is plus subscription and one is iconic subscription. So what is the difference between the two type of subscriptions? In plus subscription, you will get access to Unacademy live and recorded lectures from India's top educators. You can compete with your other learners in live tests and quizzes. Not only this, you can also get access to the exceptional question bank curated by India's top educators for you people, which contains more than 25,000 new pattern clinically integrated questions. So I can assure you, if you solve all these questions along with watching live and recorded lectures, your selection is guaranteed. Take my words. And you can download the notes, you can get printed notes also. So these are the advantages of plus subscription. In iconic subscription, along with Unacademy live and recorded lectures, you will get access to prep ladder recorded lectures also. So we know Unacademy has acquired prep ladder recently, and here you will get the benefit of both. So you can access to the best from two of the best. So if you're thinking of taking a paid subscription, better to go with iconic, because iconic have all the advantages already which are there in plus, but including that, super added to that, there is an additional advantage of prep ladder recorded lectures also. So that is the thing. So what you can do, you can check the various subscription. You can check the various subscriptions. You can go to Unacademy Learners app. You can check whether you want plus subscription or iconic subscription. You can select the duration of subscription you want, 18-month, 24-month, 36-month, various plans are available. You can check the prices of all the plans, and according to your wish, according to your need, you can select the best plan for you. And you can see longer the plan, cheaper it is. So better to go with a longer plan, but it's your wish, whatever is in your need, you can take that plan. There is an exciting news for you. If you apply my code, that is Sachdev10, Sachdev, Sachdev10 on any of these plans, whether plus or iconic, you will get straightforward 10% discount on the existing price. So don't miss out this opportunity. Okay, so let me start with today's session now. Just give me a second. Let me see. I can see your chat. Okay, I can see your chat. Okay, Dr. Fariha Salim, thanks for the compliment. Okay, so I'm starting with INICET PYQs of microbiology. We know till date only two INICETs are conducted. The first one was in November 2020 and the second one was in June 2021. So here I'm covering the PYQs of both of these of microbiology. In June 2020, we had 21 questions of microbiology and here we have 22 questions of microbiology. So I have to cover all these 45 questions of microbiology here. So today I will not be able to cover all, but 50% of them will be covered here in this lecture in the episode 1. So without wasting time, I'm coming on question number 1. So here you can see question number 1 of microbiology PYQ. See question number 1. So what is the sequence of AFB staining? What is the sequence of acid-fast bacilli staining? Now the staining is most important, a hot area from which frequently PYQs are asked. Not only in INICET, in all exams, that is NEET PG, FMGE, INICET, though it is a very important topic. So sequence of acid-fast staining is asked previous year in microbiology, but it is not necessary same the next year the same question will come. Instead of acid-fast staining, the examiner can ask you the sequence of Gram staining. Instead of that, the examiner can ask you the sequence of Albert staining. So in short, you should know all stains of microbiology. You cannot skip this portion. It is a very important area from which questions are repeated many times. Now you should understand that it is not the thing It is not the thing that the questions are repeated as such in your exams. It is the topic which is repeated. Now we know in INICET 50% of the questions will be repeat and almost 50% of the questions will be new. The repeat ones are not as it is repeat one. They are related to that topic. Now this question is from staining. Just suppose this year on this INICET you get a question of gram staining, so it will be considered as a repeat question because the topic is repeated. Now last year question was on AFB staining. This year question is on gram staining. So you should cover, my point is that you should cover the entire theory behind that. Learn the answer of this question, understand that, and आगे पीछे ऊपर नीचे उसका सब कुछ cover करो for the formation of new question ahead. Have you got it? So see the options. You can see the 4 options here. So whatever you think, you can write your option in the chat box. So most of the students are saying Akshay Kumar, Prokhar Saha, Milind. You are saying 1 as the correct answer. Okay, you all are right. So carbol fission, acid alcohol fixative, and methylene blue. So that is first option, or hematoxylin, alcohol or methylene blue second option. Carbol fission, acetic acid, hematoxylin third option. Harris hematoxylin, alcohol and eosin fourth option. So most of the students are saying 1 as the correct answer, and you all are right. Okay, the correct answer here is A. So I will give you an overview of all stains here. Just in 5 minutes, I will give you overview of all stains. Gram stain, acid fast stain, Albert stain. Then we will come on the next question. So gram stain. Why gram stain is known as gram stain? Because it is discovered by a scientist known as Christian Gram. Christian Gram in 1884, long ago, 100 years ago. So it is discovered by the scientist Christian Gram and is the most widely stain used in bacteriology. We know. We divide the bacteria based on this gram positive, gram negative. So that is the most widely stain. So our question was on acid fast stain, but you should know everything about gram stain also. So I will give you a comparative overview between the two stains. So what is the method? There are 4 steps in both stain. Gram stain, acid fast stain. In both of them, there are 4 steps. The first step is primary stain. So primary stain. Second step is mordant. You have to add a mordant. Mordant. What is mordant? Mordant is a substance which fixes the primary stain inside the bacteria. The third is decolorizer. Decolorizer. And fourth is the secondary stain. Here you use primary stain and here you use secondary stain or counter stain. Listen now. Listen now. In gram stain, the primary stain is blue in color and secondary stain is red. Here blue or violet, the primary stain. Here in acid fast stain, the primary stain is red in color and the secondary stain is blue or violet in color. That is the biggest difference. That is the biggest difference you can see between the gram and acid fast. Mordant is different in both of them. Here mordant is iodine. Here mordant is heat. And decolorizer is also different in both of them. Here decolorizer is alcohol, ethanol. Ethanol or alcohol, ethanol or alcohol. Here decolorizer is H2SO4. Especially 20% H2SO4, you have to learn the concentration also. Now compare the 4 steps. Now see, let me start with gram stain and give you the details of the gram stain. Now you will have a better understanding of this. Give me a thumbs up in the chat box if you got this. Everyone give me a thumbs up in the chat box. Okay, listen. So gram stain, let me tell you gram stain. As I have told you the 4 steps, the first step is gentian violet. It is a blue stain, or violet stain. It is a primary stain. So first you have to add any violet stain, not only gentian violet. Any violet or blue stain is the primary stain. That is your primary stain. Secondary, apply the mordant. Iodine is the mordant. What do you mean by mordant? So this is the bacteria. This is the cell wall of the bacteria, which is made up of peptidoglycan, right? And this is your blue stain. This is your blue stain or violet stain, right? That is your primary stain. So primary stain will go inside the bacteria. So then after that you are giving iodine. Then you are giving iodine. Iodine is acting as a mordant. Iodine is also going inside. Inside the bacteria, iodine will form a complex with the primary stain and keep the primary stain inside the bacteria only. Fix the primary stain there only. So primary stain cannot lose out, come out again. So that is the function of mordant. Have you got it? Give me a thumbs up. Have you got it? So mordant is iodine. The third is decolorizer. Decolorizer is alcohol, ethanol. And last is a red stain. Red is any stain. You can use carbol fission. You can use saffronin, any red stain. Have you got it? The bacteria, now there are 2 types of bacteria. The bacteria which take primary stain, they will remain violet. So these are known as gram positive. And the bacteria which take secondary stain, they will look red. So these are known as gram negative. So the bacteria, see the 4 stains. Now see the principle behind it. See the principle. Now learn a concept. The concept is that all bacteria are transparent. All bacteria are transparent. Do you know that? Yes, you know that. So can you see this is a transparent bacteria? This is also a transparent bacteria. So once you give the primary stain, that is crystal violet, all will become violet. So all here also they become violet, here also they become violet. When you give the iodine, the violet color is fixed. That is a mordant. So here they are violet, here they are violet. The third step, when you are giving decolorizer, that is ethanol. Ethanol. There are some bacteria which resist decolorization. They are not decolorized. They contain primary stain only. So they resist decolorization. And there are some bacteria which favors decolorization, they again got decolorized and they become transparent. They become transparent. Now, when you give secondary stain, counter stain, which is red in color, that is saffronin or carbol fission, then the bacteria which resist decolorization, they do not have space for secondary stain. They already filled with the primary stain. So they will remain violet only. They do not pick up the secondary stain. But the bacteria which favors decolorization, they are empty now. So they will take the secondary stain and they will become red in color. The bacteria which resist decolorization, which are violet in color, they are gram positive. And the bacteria which favors decolorization and they took the secondary stain, they are red in color, are gram negative. If you can understand this diagram, there is no confusion at all. You got the point. Give me a thumbs up. So you can see the four stains, four steps here. So once you put the primary stain, violet color on the slide, all will become violet. Then mordant, iodine, still all are violet. All are violet, all are violet. When you put decolorizer, some are violet, some are transparent. Some resist decolorization, some favors decolorization. So see, these are violet one and these are transparent one. And once you put the secondary stain, which is red in color, so the one which resists decolorization remain violet only, these are gram positive. And one which become transparent, they took the red color, these are gram negative. Everyone give me a thumbs up. Have you got it? Everyone give me a thumbs up. So here you can see both. The violet one, can you see these violet one? These are gram positive. And see these red one. The red one rods, the red color, these are gram negative. So gram positive are always violet or blue in color and gram negative are always red in color. They took the primary stain, they took the secondary stain. They resist decolorization, they favors recolorization. Any doubt? If you don't have any doubt, please give me a thumbs up. Fatafat. Iodine is a mordant that it forms a complex with the iodine dye and fixes the dye inside the bacterial cell wall. Give me a thumbs up. Now, what is the difference? As I have told you, some are resisting decolorization, some are favoring decolorization. Why? Why? It is due to the difference in the structure of the cell wall. Let me tell you. Okay. So, you can see here, this is gram positive bacteria. See the structure of the cell wall. Can you see this yellow color thing? This yellow color thing is the peptidoglycan in the cell wall. And this is gram negative bacteria. See the cell wall. See the yellow color thing in the cell wall. So you can see here peptidoglycan is more and here peptidoglycan is less. So that's why on adding ethanol जब हम add कर रहे हैं दोनों में decolorizer, so here peptidoglycan is more. So dye cannot come out. Once it is in, it cannot come out because peptidoglycan is preventing the dye to come out. That is why they are resisting decolorization. And here peptidoglycan is very less. So once you are putting ethanol, so the dye which is going in, it is coming out also. Peptidoglycan is not preventing the dye because it is very thin layer. Have you got it? So the change in the structure of the cell wall, you can see gram positive, see this purple colored peptidoglycan, and gram negative, see this purple colored peptidoglycan. So you can see here peptidoglycan is more and here peptidoglycan is less. So that is why gram positive resist decolorization and gram negative do not resist decolorization. Give me a thumbs up. Have you got it? Have you got it? So it is the structure of the cell wall which differentiate the two. So can I say, listen, can I say gram positive bacteria can become gram negative if the cell wall is damaged? If the cell, very important IBQ, very important PYQ, you know, not only in INICET, but it was a question from AIMS. So can I convert gram positive bacteria into gram negative? Yes, if I damage the cell wall. So once the damage, if I damage the cell wall of gram positive, so peptidoglycan is lost. So it will become gram negative. Have you got it? Have you got it? Have you? So that is the thing. That is the thing. So based on it, we can divide what are gram positive, what are gram negative. Based on it, total 40 bacteria are there now. Almost we have 30-40 important bacteria. So dividing on the shape of the bacteria, some bacteria are spherical, which are known as cocci, and some bacteria are rod shaped. These are known as bacilli based on the shape. Right. Now among them, which are gram positive, which are gram negative. So that is known as gram positive cocci GPC and gram negative cocci GNC. These are known as gram positive bacilli GPB and gram negative bacilli GNB. Have you got it? So there are 4 categories of the bacteria. So here you can see it is GPC, it is GNC, gram positive cocci, gram negative cocci. So ये cocci हो गए. Now coming on bacilli. that is gram positive bacilli or gram negative. You have to learn. You have to learn. Gram positive cocci are only two, Staphylococcus enterococcus, SS. Gram negative cocci only one, Neisseria. The important is only one Neisseria. So यहाँ पे SS याद करो, यहाँ पे N याद करो. यहाँ पे gram positive bacilli में there are 5, A, B, C, D, L. So actinomycetes, bacillus, clostridium, diphtheria and listeria. A, B, C, D, L. So you have to learn these 5 and remaining all, don't learn the name. Remaining all are gram negative bacilli. So this is how you have to learn. So divide in 4 categories. Gram positive cocci, gram negative cocci, ये cocci हो गए. Now coming on bacilli. Gram positive bacilli, gram negative bacilli. कैसे learn करोगे? So here SS, that is Staphylococcus streptococcus. Here Neisseria. Here A, B, C, D, L. You know, actinomycetes, bacillus, clostridium, diphtheria and listeria. And here all others. All other remaining. So this is how you have to learn. So you can crack any MCQ if you know the basics. So this is the classification based on gram staining, and gram staining is done. Coming on acid fast staining, which was our question of the previous year question in INICET, acid fast staining. So here also, why acid fast staining? What is the other name? It is known as Zeal-Nelson. Why it is known as Zeal-Nelson? Gram को gram क्यों बोलते थे? It was discovered by Gram. इसको Zeal-Nelson क्यों बोलते हैं? It is not discovered by Zeal-Nelson. It is discovered by Ehrich. लेकिन फिर भी हम इसको Ehrich नहीं बोलते हैं. It is modified by Zeal-Nelson. So it is modified by Zeal-Nelson. That's why known as Zeal-Nelson staining. Have you got it? Listen, listen. So what is the method? There are 4 steps again. The first step is the primary stain. This is primary stain. That is a red stain, which is carbol fusion. That is primary stain. Then the second step is heat. Heat is acting as a mordant. यहाँ लिखा नहीं है, you can add mordant, mordant, mordant. So it is a mordant. वहां पे iodine था, यहाँ पे heat है. Third is decolorizer. Decolorizer वहां पे था ethanol. यहाँ पे 20% H2SO4, 20% sulfuric acid. Then secondary dye or counter stain, which is a blue stain or violet stain, that is methylene blue. तो primary, secondary उल्टे पुल्टे हो गए. It is flip flop. Have you got it? Have you got it? The organism which take primary, they are red in color. They are acid fast organism. And the organism which took secondary, they are blue in color. These are known as non-acid fast organism. तो वहां पे gram positive, gram negative थे. यहाँ पे acid fast और non-acid fast है. यहाँ पे positive, negative नहीं है. That is how you divide. Have you got it? Have you got it? Let me see. I can see your chat. Okay. I can see your chat. Okay. Have you got it? Have you got it? So these are the 4 steps you can see. Initially, all bacteria are transparent. See, all of them are transparent. You know, all bacteria are transparent. This is the punchline. So whenever you are giving the primary stain, carbol fusion, that is a red stain. So all will become red. See, this is also red. This is also red. All will become red. Whenever you heat, then again it will fix it. So all are red, all are red. Then you are adding decolorizer, 20% H2SO4. This step, now some of them resist decolorization, they will remain red only. And some of them favors decolorization, they will lose the red stain and they become again transparent. They will become transparent. Now when you add the counter stain, secondary stain, which is a blue stain, then the one which resists decolorization, they do not have space inside it. So they will not take the secondary stain. They will remain red only. They will take primary stain only, right? The one which become transparent, which favors decolorization, they are empty now. So they will take the secondary stain, that is a blue stain, and they will become blue in color. So the one which are red, which resist decolorization, which took primary stain, these are known as acid fast organism. And the one which favors decolorization, which loses the primary stain and take the secondary stain, they look blue in color. These are known as non-acid fast organism. It is not gram positive and gram negative. It is known as acid fast and non-acid fast. Give me a thumbs up. So the same concept is shown here also. The red one are acid fast and the blue one are non-acid fast. Right? They took the primary stain because they resist decolorization. They took the secondary stain because they favors decolorization. Now why it is happening? What is the principle behind it? वहां पे principle क्या था? The peptidoglycan layer in the cell wall. Cell wall में peptidoglycan ज्यादा है तो it is resisting. और वहां पे peptidoglycan कम है तो it is favoring. यहाँ पे the thing is mycolic acid in the cell wall. It is not peptidoglycan. It is mycolic acid. So the concept is clear to you. The acid fast are one which are red in color and the non-acid fast are one which are blue in color. So see here. Can you see the red color organisms? Please appreciate these red rods. All of them are acid fast organisms, because these are red in color. They are retaining the primary state because they are resisting decolorization, because they have mycolic acid in their cell wall. See these red rods, red. Please appreciate, they can come for your IBQs also. So IBQs bhi solve karte jao saath saath mein, right? Okay. What is the principle behind it? What is the principle behind it? Okay, let me tell you the principle. The principle is mycolic acid in the cell wall. Can you see here? Okay, let me draw a diagram for you. Okay, so this is a bacteria. This is a cell of the bacteria. This is the cell wall. Now, only few bacteria out of the 30-40 bacteria, only few of them have mycolic acid. Mycolic acid, it is a lipid. It is a lipid. It is present in the cell wall. Not all bacteria, only few of them. So once you give the primary stain, that is red in color. So once you give the primary stain, so this mycolic acid is preventing the decolorization. Because of mycolic acid, this primary stain cannot come out and it will remain inside only. So it resists the decolorization because of mycolic acid. So all those bacteria which have mycolic acid in their cell wall, they are acid fast organism, and all the bacteria which do not have mycolic acid, they have peptidoglycan in their cell wall, they are known as non-acid fast. So out of the 40 bacteria we are dividing. So what is fastness? Fast का मतलब not speed, slow and fast वाला fast नहीं है. Fast का मतलब होता है resistance. What is the decolorizer? Decolorizer is an acid. It is 20% H2SO4. So the bacteria which resist acid are known as acid fastness. So acid fastness means resistance. It is the resistance. Have you got it? Fastness is the resistance. So out of the 40 bacteria, only 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 of them have mycolic acid in their cell wall. So that's why these are acid fast. Remaining all are non-acid fast. So non-acid fast का list learn करने की जरूरत नहीं है. You have to learn the list of acid fast. Have you got it? What is the list of acid fast? So there is a mnemonic for you. So mnemonic is no longer separate booking for RIM SIM card. So no ka nocardia, longer ka L is legionella, S separate ka S magma, booking ka B is bacterial spores, RIM, R is rhodococcus, isospora, mycobacterium, RIM, mycobacterium both, tuberculosis also, leprosy also, and SIM ka S is sperm, male sperm, human sperm, card ka C is cryptococcus. So these are the 9 organisms or structures you can say which are acid fast organisms. Otherwise all are acid negative. Give me a thumbs up if you got it. Give me a thumbs up. So this is a mnemonic. Now out of them, some of them are bacteria, some of them are parasites, and some of them are others. You can separate them. इनमें से बैक्टीरिया कितने हैं? माइकोबैक्टीरिया, नोकार्डिया, रोडोकोकस, मैग्मा एंड लेग्यूनेला. सी इनमें से बैक्टीरिया 5 हैं. इसमें पैरासाइट्स हैं, क्रिप्टोस्पोरा, साइक्लोस्पोरा, आइसोस्पोरा, और अदर्स में स्पोर्स हैं और स्पर्म है. सो यू कैन डिवाइड लाइक दिस ऑल्सो कि इनमें से बैक्टीरिया कितने हैं बताओ, इनमें से पैरासाइट कितने हैं बताओ, अदर्स में क्या क्या आएगा बताओ. जो ना बैक्टीरिया है ना पैरासाइट है, दैट इज स्पर्म, स्पोर्स. So like this. So you can divide like that also, like this also. So that is the thing. Have you got it? Have you got it? There are some modifications in Ziehl-Neelsen stain. That is known as modified Ziehl-Neelsen. What you have to learn here, in modification, normally we use 20% H2SO4 as decolorizer. Instead of 20% H2SO4, if I use 5%, 1%, 0.5% and 0.25%, it is known as modified Ziehl-Neelsen. So you have to learn. We use 5% for Mycobacterium leprae. We use 1% for actinomycetes. We use 0.5% for nocardia. And we use 0.25% for cryptospora, isospora and bacterial spores. Otherwise, by default, we use 20%. So there are many MCQs which are asked on this modification, especially leprae. 5% बहुत बार PYQ में आ चुका है. Not only INICET, in NEET PG, AIMS, PGI, JIPMER, NIMHANS. So you should know PYQs of all exams, FMGE. Not only INICET. ऐसा नहीं है कि मेरा टारगेट तो INICET है तो मैं तो सिर्फ वहां के PYQ करूंगा या करूंगी. It is not the thing. If you are appearing in any exam, you should do past 5-year MCQs of all possible exams, right? Especially of that exam which you are appearing. But you should do of all other exams also. So this is a favorite PYQ of the examiner, you know. So you should know. So out of these, इनमें से यू सी नोकार्डिया 0.5%, तो यहीं पे लिख दो 0.5% नोकार्डिया को, 0.5% नोकार्डिया. एक्टिनोमाइसिटीज इज 1%. Okay, in this mnemonic I have not included actinomycetes actually. And cryptospora, isospora and spores. This is isospora. This is bacterial cryptospora. and this is bacterial spores. ये तीनों आपके 0.25 हो जाएंगे, 0.25, 0.25 and 0.25. Okay, so let me tell you, mycobacterium is of two type, TB and leprosy. इनमें से जो leprosy वाला है वो हो जाएगा आपका 5%, 5%. तो एंड अदरवाइज जो बच गए, जहां जहां जो बच गए, दिस वन जो भी बच गए वो सब आपके 20%. So you can learn like this. Give me a thumbs up, give me a thumbs up, give me a thumbs up. That is the thing, that is the thing. So can you compare the two stains now? What are the two stains? So gram stain, acid fast stain. Gram stain, acid fast stain. The first question is over. So tell me the primary stain for both of them. Tell me the mordant for both of them. Tell me the decolorizer for both of them, and tell me in the end, tell me the secondary stain or counter stain for both of them. Who will tell me? You can write your answer in the chat box if you know the answers. For gram stain, what are the correct answers? So primary stain is a blue stain here, and here it is a red stain, right? Mordant here is iodine, here is heat, right? Decolorizer here is alcohol, ethanol, and here is 20% H2SO4 by default, and you know the modifications. Secondary stain यहाँ पे red है और यहाँ पे blue है. I guess the concept is clear to you. Give me a thumbs up. The second thing, based on which, what is the classification? What is the classification? So here we divide into 4 categories: gram positive cocci, gram negative cocci, gram positive bacilli, gram negative bacilli. And here we divide in 2 categories only: acid fast organism, non-acid fast organism. So you know the classification. Give me a thumbs up if you know the classification. Everyone give me a thumbs up if you know the classification. So gram positive cocci, gram negative cocci, gram positive bacilli, gram negative bacilli. ये 4 क्लासिफिकेशन ग्राम स्टेन के हैं. और एसिड फास्ट ऑर्गेनिज्म और नॉन एसिड फास्ट ऑर्गेनिज्म, ये 2 क्लासिफिकेशन आपके उसके हैं. दैट इज एसिड फास्ट स्टेन. हैव यू गॉट इट? तो यहाँ पे स्टेफाइलोकोकस, स्ट्रेप्टोकोकस, यहाँ पे निजेरिया, यहाँ पे ABCDL, यू नो द फुल फॉर्म. यहाँ पे ऑल रिमेनिंग. डोंट लर्न द लिस्ट. गिव मी अ थम्स अप. यहाँ पे देयर इज नो सेपरेट बुकिंग फॉर सिम card. You know the full form, and यहाँ पे all remaining, and you know the classification, you know the full forms of this. Everyone give me a thumbs up, everyone. So let me come on the next question. Are you people there? Please reply me by giving a thumbs up in the chat box so I can understand you got it. If you are listening, if I'm audible, if I'm visible, and if you understood the concept in the first question. So let me read the second question now. Okay, thank you. Let me read the second question now. Here is the second question for you people. Okay, it is a difficult question. It is a question from June 2021. So pneumonia due to gram negative. What is the hints given in the question? The first question, the disease caused by the organism is pneumonia. It is a gram negative pleomorphic bacilli. It is not cocci. It is a bacilli. It is not gram positive. It is gram negative, and it is pleomorphic. Now I have told you the classification. Now I have told you the classification, and it is growing only on chocolate agar, and it is catalase positive, oxidase positive. So name the organism. जिसमें ये सारी properties हैं, जिसमें ये सारी properties हैं. All these properties. Tell me, कौन सा ऐसा bacteria? There are 4 options: E. coli, Pseudomonas, Haemophilus influenzae, Actinomycetes. I will give you 10 seconds to write the answer in the chat box, right? So please read the question. Tell me out of these four options, tell me the answer which is having all these properties. What are the properties? Number 1, it should cause pneumonia. वो organism pneumonia कराना चाहिए. Number 2, it should be gram negative, not gram positive. Number 3, it should be pleomorphic. It should change. It can change its shape frequently. It should be a bacilli. It is not cocci. It can grow on chocolate agar. It is catalase positive. It is oxidase positive. Now, I have taken a separate class for the micronemonics in which I have given you several mnemonics. If you can see that special class, free class are available on Unacademy platform. Please watch that class before going to your exams, right? I have given you a mnemonic, all those bacteria which are catalase positive. Name the bacteria. All those bacteria which are oxidase positive. Name them. So there is a list which is common for catalase and oxidase. I have given that list also. I have given the list. अभी मैंने आपको बताया ग्राम नेगेटिव कौन से हैं. अभी भी मैंने बताया बेसिलाई कौन से हैं, कोकाई कैंसर. सो यू शुड कंबाइन ऑल द लिस्ट. उनमें से एक आंसर निकलेगा. यस, एनीवन? सो एवी सोनी इज सेइंग सी एज द करेक्ट आंसर. सी इज द करेक्ट आंसर. यस, आई गेस यू आर राइट. एनीवन एल्स अपार्ट फ्रॉम इट? Anyone else apart from it? Yes or no? Anyone else want to try? Give me a thumbs up. Okay. So let me tell you how to approach. Approach is important. How to approach this question? Okay, so C, answer is C. As A.V. Soni have told us, the answer is C. But how, A.V. Soni, how you came to this answer? So actinomycetes is a gram positive bacilli. I have told you na, A, B, C, D, L was the mnemonic. Just मैंने पिछले क्वेश्चन में बताया. तो A, B, C, D, L का A क्या है? It is actinomycetes ना. It is a gram positive coca है. The question में gram negative coca है पूछा है. ग्राम नेगेटिव पूछा है, तो एक्टिनोमाइसिटीज तो आंसर नहीं हो सकता ना. इट इज ग्राम पॉजिटिव bacilli. In the question, gram negative bacilli is asked. So this option is ruled out based on this property. It is not gram negative, it is gram positive. तो ये तो हटा दो. चलो अगले पे आते हैं. E. coli is oxidase negative. We know it is oxidase negative ना. Oxidase negative की लिस्ट में आएगा E. coli. It is not oxidase positive. तो ये ऑप्शन भी हटा दो because it is oxidase negative and we want a bacteria which is oxidase positive. Catalase positive, oxidase positive both. तो ये भी हटा दो. And now we are left with two options: Pseudomonas and Haemophilus. Both of them are oxidase positive, both of them are catalase positive, both of them are gram negative bacilli, right? तो ये properties इन दोनों में, both of them are gram negative bacilli, both of them are oxidase positive and catalase positive. अब आप बताओ इनमें से चॉकलेट अगार पे कौन ग्रो करेगा और इनमें से प्लियोमॉर्फिक कौन है? इन दोनों प्रॉपर्टीज की बेसिस पे यू विल कम ऑन द आंसर. हैव यू गॉट इट? सो Pseudomonas and H. influenza both are gram negative bacilli. Okay, both of them are catalase positive, both of them are oxidase positive, but it is the Haemophilus influenzae which has other two properties, that it can grow only on chocolate agar because it requires specific growth factors, that is factor 10 hemine and factor 5 for NAD, without which it cannot grow. And both of these are present on chocolate agar, heated blood. When you heat the blood, these factors will come out. So it is the H. influenzae which grows only on chocolate agar. Pseudomonas zaruri nahi hai chocolate agar pe hi grow karo. Have you got it? Number two, H. influenzae is also showing considerable pleomorphism, and Pseudomonas do not show pleomorphism. So all five properties are fitting in H. influenzae. So that is how you should approach this question. That is why the correct answer here is H. influenzae and not other. Pneumonia तो ये सभी करा सकते हैं. Mostly all of them cause pneumonia. तो pneumonia को मैंने consideration में लिया ही नहीं. Give me a thumbs up. Give me a thumbs up if you got my approach. I tried hard. So it is how you have to approach the questions, the clinical questions in bacteriology. So not only this, you should practice other clinical questions also. So I am launching a crash course for bacteriology in which I will cover all important 40 bacteria from which frequently questions are asked in just 5 hours. So it is a 5-hour marathon in which I will cover entire bacteriology. So I'm launching this series in the next week. So don't miss it out. Don't miss it out. I will take it in 2 parts, 2.5 hours. Total 5 hours ka session hoga. That is bacteriology crash course for the upcoming exams, right? So don't miss it out. Anyways, let me come on the next question. Let me come on the next question. See this question. I'm sorry. Please read the question and tell me what is the correct answer. Please read the question and tell me what is the correct answer. Okay. So see, I will read the question for you. So there is a patient who presents with mobile warm to touch nodule in the neck. So patient is having a nodule in the neck somewhere. The nodule is mobile. On anaerobic culture of the biopsy material from the swelling, we have taken a biopsy and the biopsy is grown on an anaerobic culture. It is forming a colonies as shown in the figure. See the colonies. I'm really sorry. I'm really sorry. Please see the colonies. There is a specific thing given in this colony. Please appreciate that. How the colonies are given. And there is one more hint. It is a gram positive branching rod bacteria on staining, not on culture, on staining. If you stain it, it is not gram negative, it is gram positive. It is not cocci, it is bacilli. But it is branching bacilli like fungus. Like fungus, it is branching bacilli. It is branching. So first give me the diagnosis. A.V. Soni, you are saying the answer C. You are correct. Penicillin. First tell me diagnosis क्या है? ट्रीटमेंट पे बाद में आएंगे. ट्रीटमेंट ड्रग ऑफ चॉइस फॉर दिस कंडीशन. डायग्नोसिस तो बताओ कौन सा बैक्टीरिया है और कौन सी डिजीज है. 2 क्वेश्चंस A.V. Soni. A.V. Soni, 2 questions for you. So what is the diagnosis? Which bacteria we are talking about? So see the diagram. See the colonies. See the shape of the colonies. Can you see white colored colonies shaped like this? So it is looking like a molar tooth. We have 4 types of tooth. Na? Canine, we have incisors, we have canine, we have molar, we have premolar. So molar कैसे होते हैं आपके? It is like this. So this is the molar tooth. So this is typical shape of a molar tooth colony. Molar tooth colony on anaerobic culture. आपको आंसर बताओ? Yes. So Yash, AB Soni, you all are right. The answer is actinomycetes. The typical appearance is typical. It is a spotter actually. It can come for IBQ also. अब जरूरी नहीं है next year question यही का यही आए. In this upcoming exam, you can get this IBQ. Identify the colonies किस organism की हैं. So it is typical molar tooth, or give a typical name to this colony. The name to this colony is molar tooth colony. It is seen in actinomycetes, right? So molar tooth appearance, it is a gram positive branching rod also. वो भी fit हो रहा है. So it is a gram positive branching rod also, and it is a molar tooth colony. Both the criteria are fitting. So the answer is nothing else. It is actinomycetes, or actinomycetes का drug of choice is penicillin. Drug of choice is penicillin and tetracycline. अगर इसे ठीक नहीं होता है तो we will go for surgery. Have you got it? Have you got it? So here penicillin is the correct answer. अगर ऑप्शन में penicillin ना हो और टेट्रासाइक्लिन हो तो गो विद टेट्रासाइक्लिन, और वो भी ना हो उन दोनों के अलावा फिर सर्जिकल ड्रेनेज. फर्स्ट वी विल ट्राई विद एंटीबायोटिक्स. एंटीबायोटिक्स वर्क नहीं करेंगे तो वी विल गो विद सर्जिकल ड्रेनेज. हैव यू गॉट इट? सो आई एडवाइज यू, इस क्वेश्चन से हमें क्या सीख मिली, what lesson we have learned from this question? All important colony appearance of all 40 bacteria you should know. किसमें क्या स्पेशल, नो पर्ल ऑफ स्ट्रिंग कॉलोनी किसमें होता है, व्हाइट पर्ल्स किसमें होता है, कौन सा कॉलोनी स्पेसिफिक कलर किसमें होता है, गोल्डन कलर कॉलोनीज किसमें होता है, ग्रीन कलर कॉलोनीज किसमें होता है, एंड डोनट अपीयरेंस कॉलोनीज किसमें होता है. सो यू शुड नो कॉलोनी अपीयरेंस ऑफ ऑल इंपॉर्टेंट 40 बैक्टीरिया. So I will take that crash course now, which I am talking about. I will cover all important colony appearance. So that can help in your IBQs also and in your clinical questions also. Have you got it? So first, looking at the colony appearance, give me the diagnosis. Once the diagnosis is there, you know the treatment, you know the drug of choice, Give me a thumbs up. The correct answer here is penicillin, because the diagnosis is actinomycetes. The diagnosis is based on the colony appearance. It is a molar tooth. Now, instead of this image, same question. इमेज मैं दूसरा दूंगी. इमेज में ये कल्चर का है, एनारोबिक कल्चर का स्टेन ना देके मैं ग्राम स्टेनिंग का इमेज दे दूं तो सेम क्वेश्चन, सेम ऑप्शंस, एबीसीडी सेम ऑप्शन, सेम स्टोरी, सेम क्वेश्चन. दिस इमेज की जगह मैं ये वाला इमेज दे सकती हूँ तुम्हें. आई कैन गिव यू दिस इमेज. व्हाट इज दिस इमेज? नाउ सी द ऑर्गेनिज्म, वेदर दे आर रेड कलर की ब्लू कलर. If they are blue color, they are gram positive. If they are red color, they are gram negative. I have told you in the first question. See, these are the organisms. So which color organisms you can see? All of them are blue color. So of course they are gram positive, and they are bacilli. They are long, they are rods, they are not cocci. So gram positive bacilli. ग्राम पॉजिटिव बेसिलाई 5 ही होते हैं, ए बी सी डी एल. तो इन पांचों में से ही तुम्हारा आंसर होगा. There is one more hint given in this question. See the bacteria, they are branching. Can you see it is branching like a fungus. So of course the answer is A. The answer is A, that is actinomycetes. So instead of this image, you can get the same question with this image also. So answer will be same. Instead of these 4 options, you can get the name of 4 bacteria. What is the answer? So 4 bacteria भी आ सकते हैं, तो कुछ भी question आ सकता है related to the same story. Have you got it? You should be prepared for all. Give me a thumbs up. Everyone give me a thumbs up. So what are actinomycetes? Actinomycetes are of 2 types. You know the story. They are nocardia and actinomyces. So these are the 2 types of actinomycetes. Both of them are gram positive bacteria that resemble fungus because of the branching pattern. That is known as mycete का मतलब होता है fungus. And they form an extensive colony which is known as mycelium. And both of them cause granulomatous disease, right? They are actually not fungus, but they look like fungus, right? See the colonies. Colonies are looking, see the colonies, they are looking like molar tooth appearance on culture, and on gram staining they are forming branching blue color rods. So branching blue rods, that is gram positive rods. So see the typical appearance on culture, see the typical appearance on gram staining. Any image can come, either this or this in your exam. The answer is same, actinomycetes ना देके ऑप्शन में nocardia दे दिया तो go with that. Nocardia is a type of actinomycetes only. Give me a thumbs up. So I tried to cover all possible other questions based on this question. I have solved this question and all other possibilities which can come in your exam based on this question. We have covered that also. Give me a thumbs up. Fatafat, give me a thumbs up so I can move to the 3rd question, 4th question. Can I move to the next question? Are you people there? Give me a thumbs up. Okay. So this is the next question in front of you. Again, this is the question from June 2021. INICET micro. You can see, I will read the question for you. Which protein is present in HPV vaccine? Which protein is present in HPV vaccine? You know, human papilloma virus vaccine. So what is human papilloma virus? It is the oncovirus. You know, it is the oncovirus. So it can cause cancer. So which cancer? If it is a male and it is a female, it causes gonadal cancer, genital cancer. In males, it causes penis cancer, and in females, it causes cervix cancer. These are the malignant cancers. These are the malignant cancers caused by this. It also causes benign cancers. Benign cancers. The name of the benign cancers is warts in both. In male also, in female also, it causes warts, the benign cancer. So in short, in benign cancer, it causes warts. And in malignant cancer, it causes genital cancer. That is penis cancer in males and cervix cancer in female. So it is a oncovirus, so it causes cancer. So it is a very dangerous virus. It is a very dangerous virus. Once it enters the human body, it will cause certain mutations. Which mutation? You have to know the name of the mutation also. The two genes are mutated, P53 and retinoblastoma. Because of mutation of both of them, these cancers are called by HPV virus, right? So it is how HPV virus is oncogenic. So we have a vaccine for it. We have a vaccine. Scientists have developed vaccine for it. So vaccine में कौन सा प्रोटीन होता है? कौन सा E6 E7 प्रोटीन होता है कि L1 प्रोटीन होता है कि E1 E2 प्रोटीन होता है कि L2 प्रोटीन? So vaccine contains a attenuated virus. Like COVID virus, we use attenuated virus. It is a what do you mean by attenuated virus? It is a live virus. It is not dead, but it is non-pathogenic. It cannot cause disease. It cannot cause cancer. It can only provide immunity. So it is immunogenic but non-pathogenic. The immunogenic but non-pathogenic thing is known as attenuated, right? So we use the attenuated virus. Actually virus का we use specific antigen. So actually इनमें से कौन सा antigen है, कौन सा प्रोटीन है which causes cancer, जिसके अगेंस्ट हम वैक्सीन बनाते हैं? Have you got it? So the answer, anyone have any guess? Most of you are right. Most of you are right. Yash, Kaushik, Harshita, AV Soni, Ikwar, everyone is right. The correct answer here we all know is L1. But we should know the details. Answer चलो इसका तो L1 सबको पता है because it is a PYQ and you have already solved the PYQs. So see, this is the structure of a virus, HPV virus, and this is the genetic material of the virus. Inside this virus, there is a protein which is known as L1. When this virus enters the human cell, it is L1 which causes mutations. And because of this L1, because of the mutation, there is cancer. So when we prepare the vaccine, we take this L1 protein which is attenuated. It is live but it is attenuated. So our body will form antibodies against this L1, and when actual virus will come in the body already have antibodies against the L1. So it is the L1 which is oncogenic. That's why vaccine contains L1. How many types of vaccine you know? HPV virus की कितनी तरह की? No, it is not E6. Thakir Bapu, no, it is not E6. The answer is L1, right? L1 is the non-infectious recombinant virus-like particles composed of HPV L1 proteins vaccine में होता है. How many types of vaccines are there? Now question इस पे भी नहीं आ सकता, जरूरी नहीं है the same question is repeated. Question can come on the type of vaccines. There are 3 type of vaccines which are approved by FDA for HPV. I hope you all are vaccinated for that. If you are not, you can go because you are in specific age group, I guess. Anyways, there are 3 types of vaccine: bivalent, quadrivalent, and 9-valent. So what is the name of these vaccines? Bivalent vaccine is known as Cervarix. That is a company name, brand name, right? So that is the name. Like अभी हमारे पास Pfizer का वैक्सीन है कोविड के लिए, you know, Moderna का है. We have various types of vaccines. यहाँ पे Cervarix है. There is Gardasil, and there is Gardasil 9. Gardasil, it is quadrivalent. So we have HPV is of more than 100 types. More than 100 types of HPV is there. So more than 100 types में से 6 and 11 causes benign warts, benign tumor, that is warts. 16 and 18 causes malignant cancers, that is penis cancer in males and cervix cancer in females. तो तुम्हारे लिए क्या जरूरी है? वैक्सीन में क्या प्रिवेंट हो? कौन सा वाला वैक्सीन यू वांट स्पेशली दिस बिनाइन चलेगा लेकिन मैलिग्नेंट नहीं होना चाहिए. सो वैक्सीन शुड कंटेन the attenuated virus 16 and 18. So bivalent contains 16 and 18. So malignant का तो prevention हो गया. Quadrivalent along with 16, 18 के अलावा 6 और 11 भी हो गया. और 9 valent में 6, 11, 16, 18 के अलावा 5 और आ गए. So that is क्या क्या आ गए आपके 33, 45, 52, 58. Have you got it? 31, 32. So total ये हो गया. So you can see here. You can see here. So it is, there are थ्री टाइप ऑफ वैक्सीन किसमें, हियर 16 18, यहाँ पे 16 18 6 11, यहाँ पे 16 18 6 11 31 33 45 52 and 58. You have to learn this number. So it is bivalent because only 2 HPV viruses are covered. Here quadrivalent because 4 viruses are covered, and here it is nonavalent because 9 viruses are covered. So you have to learn the numbers. You have to learn the numbers. Bivalent में कौन से दो हैं? 16 18 because they cause malignant. तो malignant तो prevent हो गया, लेकिन benign हो सकता है. The one which is vaccinated for this one, उसको malignant नहीं होगा, लेकिन warts हो सकते हैं. The one which is vaccinated for this one, quadrivalent 16 18 6 11, उसको न तो benign होगा न तो malignant होगा. लेकिन ये चार most common है, उसके अलावा others भी हैं. The one which is vaccinated for this one 9 वाला, उसको very less chances कि benign भी होगा और malignant भी होगा, उसको कुछ भी नहीं होगा. So that is the thing. So these are the vaccines. These are the vaccines. The bivalent is approved only for females, but the quadrivalent and nonavalent is approved for males and females both, right? So bivalent सिर्फ females की हैं. Age group is same. That is 9 to 25 years. 9 to 25 or 26 years के लिए approved है. If you belong to this age group, go and get yourself vaccinated. If you are a female, you can get vaccinated for any of them. But if you are a male, you cannot vaccinate it for bivalent. You have only two options, quadrivalent or nonavalent. So if nonavalent is available, that is the best. It is costly, but it is the best. Otherwise, go with quadri. If you can't afford quadri and it is not available, go with bivalent. So bivalent सबसे best तो nona है. Have you got it? Give me a thumbs up, everyone. Everyone, give me a thumbs up, everyone. Have you got it? So that is the thing. You can see bivalent vaccine and quadrivalent vaccine. तीसरा nona भी आ गया है अब. So you can see the duration. You can see the regimen here. So three times you have to be vaccinated for complete vaccination. So if you are vaccinating today, after 1 month and after 6 months. If you are vaccinating today, after 2 months and 6 months, that is the schedule. That is the schedule. You have to do it for preventing cervix cancer in female and penis cancer in males. Have you got it? That is the thing. Give me a thumbs up. Should I come to the next question? Should I come to the next question? Just a second. Give me a thumbs up in the chat box. Yes, it is condyloma acuminata. Yes, right. That is the benign lesion. That is the name of the warts you are saying. Right. Should I come to the next question? This is the next question. It is a difficult question. I will give you all the details. Please read the question. The question is based on the pathogenic variants of E. coli. We know there are 5 pathogenic variants of E. coli. ETEC, EIEC, EHEC, EPEC, and the 5th one is also there. So tell me, match the column. You have to match the column. You have to tell me the mechanism of their virulence or pathogenicity. Now the 4 options are in front of you. What are the 4 options? Which one among them produce a stable, heat stable toxin and causes the activation of guanylyl cyclase and produces cyclic GMP? Which one among them invades intestinal mucosa? Which one among them produces a toxin against vascular endothelial cells? And which one among them do not invade but adheres to the mucosa and affects the microvilli of the intestine and causes the toxin? Now compare. These are your options. Now you have to match it. A को match करोगे 2 से, 3 से, 4. So whatever you wish, you can do it. I will give you a few seconds. I will give you 15 seconds. It is a difficult question. Everyone try it out. Write your answer in the chat box. Then I will teach you all 5 types of E. coli. Then I will come on the question again so that your concept is clear. Give me a thumbs up. It is a very important topic. Very few students know the mechanism of action of various types of E. coli pathologically, pathogenicity ke basis pe. Give me a thumbs up. Give me your answer. Most of them are saying answer is B. I don't know the correct answer. Let me see. AB Soni Harshita is saying answer as B. What about others? What about others? Write your answer in the chat box. Write your answer in the chat box. No, you are wrong. Those who were saying B as the correct answer here is C. Okay, I will come on the question again. Don't worry. अभी मैं C पे जाके एक explain करूँ, वो कुछ मतलब नहीं है. वो कुछ मतलब नहीं है. Let me tell you the 5 types of E. coli. Then I will come on this slide again. I will move back and come on the slide again. So let me teach you the 5 types of E. coli. Okay, E. coli. How many diseases E. coli causes? E. coli क्या क्या करवाता है? What are the diseases E. coli causes? E. coli causes 4 types of diseases. It causes UTI, it causes diarrhea, it causes pyogenic infection, it causes septicemia. I am interested in diarrhea. For diarrhea, based on this property, there are 5 types of E. coli. Okay, I will teach you a little bit about the UTI and then I will come on the diarrhea. Okay, So UTI, it causes UTI, it causes diarrhea, it causes pyogenic infection, and it causes septicemia. For causing diarrhea, there are 5 different types of E. coli. These are the 5 different types of E. coli. I will tell you the names. First, let me tell you UTI. UTI. So UTI is the most common, E. coli is the most common organism which causes UTI in humans. So what are the frequent serotypes of E. coli which causes UTI? O1, O2, O4, O6, O7, O18 and O75. These are the O. You know, there are 2 types of antigen, O and H. So these are the somatic antigens. So these are the various serotypes of O which causes UTI. And some of the strains causes nephrogenic strains also, which are specific for causing UTI. Have you got it? Okay. How E. coli causes UTI? For causing UTI, the E. coli should reach the urinary bladder. This is the kidneys. These are the 2 kidneys. These are the 2 ureters. This is bladder and this is urethra. So how for causing UTI, the E. coli should reach the kidney ना, kidney तक पहुंचेगा कैसे? ये E. coli bacteria, what is the route? E. coli कैसे पहुंचेगा kidney तक? Once it reaches the kidney, it will cause UTI. It will cause UTI there, urinary tract infection. So there are 2 routes. Listen, ascending route and hematogenous route. What is ascending route? The most common route is ascending route. So can you see urethra? So the bacteria is going inside from the urethra. यूरेथ्रा से जाएगा ब्लैडर में, ब्लैडर से जाएगा यूरेटर में, यूरेटर से जाएगा किडनी में. दैट इज नोन एज असेंडिंग रूट. हैव यू गॉट इट? तो यूरेथ्रा में कहां से आएगा? यू नो, इन ह्यूमंस देयर आर 2 ओपनिंग. In humans, this is urethral opening from which urine comes out, urethral opening, and this is anal opening from which feces come out. Now anal opening, here there is colon or rectum leads to anal opening. So E. coli is the commensal for anal canal. E. coli इसमें normally होता है. We all have E. coli in our colon, but it is a commensal. It is not a disease. But if E. coli is present in urethra, it is a disease. So E. coli is a commensal in the anal canal, but not in the urethral canal. And the 2 openings are close to each other. So due to the vicinity of the 2 openings, the E. coli from the anal canal, they can travel here and it can enter into the urethra and it can ascend upwards into the urethra. Have you got it? So this is known as ascending infection. What is hematogenesis? Through blood. You know, this is aorta. You can see the aorta, and these are the renal artery. If blood is present, if bacteria is present here in the blood, it can enter the kidney. That is hematogenous. So the 2 routes are ascending and hematogenous. Ascending is more common. It is fecal contamination of the urethra. Have you got it? So we can say it is an endogenous infection. Now ascending route is more common in females. Why? Because in females the urethra is short. You know, in females this urethra is very short in females, so it can easily ascend. In females, urethra is short. Number 2, due to sexual intercourse, sometimes the females have injury over the urethra, and in injured region, bacteria especially comes in the injured region. Yes, so that is the thing. So that is known as honeymoon piolitis. Number 2, due to the sexual intercourse. And number 3, females do not have prostate. Males have prostate. So prostatic secretions are present in the urethra which kills the bacteria. But in females, the prostate gland is absent, because of which again UTI is more common in females. So we always know UTI is more common in females. But why? E. coli causing UTI in females is more common, not in males. There are 3 reasons: shorter urethra, absence of prostate, and urethral trauma during sexual intercourse, that is known as honeymoon piolitis. Give me a thumbs up. No. And second is hematogenous route via blood. That is the thing. I'm not interested in UTI because our question is based on diarrhea. Let me teach you diarrhea. For causing diarrhea, there are 5 types of E. coli which have different pathogenic mechanism. All of them causes diarrhea, but different mechanism. Let me teach you the 5 mechanism. Let me teach you the full form of them. EPEC is enteropathogenic E. coli. Pathogenic से P है. ETEC is enterotoxogenic E. coli. यहाँ से पैथोजेनिक था, यहाँ पे टॉक्सोजेनिक है. EIEC is enteroinvasive. ये पैथोजेनिक, ये टॉक्सोजेनिक, ये इन्वेसिव. Enteroinvasive E. coli. The 4th one is enterohemorrhagic E. coli. इसको VTEC भी कहते हैं कुछ लोग in some books. It is known as virocytotoxic E. coli. And last is EAAC, A for aggressive or adherence. Enteroaggressive E. coli. So what are the 5 types? Tell me. So EPEC, ETEC, EIEC, EHEC, EA. So enteropathogenic, enterotoxogenic, enteroinvasive, enterohemorrhagic, enteroaggressive. First give me a thumbs up if you got the names. So let me tell you the 5 different mechanisms of diarrhea. Let me tell you the 5 different mechanisms of diarrhea in each of them. Let me start with the first one, EPEC. Let me start with the first one, EPEC. So it causes diarrhea, especially in infants and children, right? Okay. So what is the mechanism? It does not produce a toxin. It is non-invasive. So what it does, it adheres to the mucosa of the small intestine and disrupts the microvilli. Disrupts the villi are present. So it causes degenerative and inflammatory changes in the microvilli, which causes diarrhea. Okay, let me draw a diagram for you. You can see this is esophagus, this is stomach, and this is small intestine. This is the small intestine. So this is the lining of the small intestine. Let me draw the lining, few cells in the lining of the small intestine, and the small intestine lining have microvilli. This is microvilli. This is microvilli. So if EPEC is present, let me draw the bacteria. This is EPEC. EPEC. Now whether it produces toxin? No. It is going inside the cell? No. It is just adhering to this microvilli. It is just adhering to this microvilli and destroying these microvilli. If these microvilli is destroyed, the patient will have diarrhea. So that is the mechanism of action, how it acts. Give me a thumbs up. Give me a thumbs up. So that is the thing. Everyone give me a thumbs up. Everyone. So mechanism of action of EPEC, it adheres to microvilli and destroy them. It do not produce toxin. It do not invade inside the cell. It remains superficial only. It just adheres superficially and destroy, cause degenerative and inflammatory changes in the microvilli, which causes diarrhea. Let me come on the second, ETEC. ETEC. Give me a thumbs up. ETEC. As the name indicates, toxogenic, so it will produce toxin. It is the most common cause of diarrhea in traveler's diarrhea, in traveling persons. It causes traveler's diarrhea, right? Traveler's diarrhea. Again, it is non-invasive. ये अंदर नहीं जाएगा. It doesn't adhere. ये खुद adhere नहीं करेगा. Like the previous one was adhering on the microvilli. It will produce toxin. It will produce two types of toxin, LT or ST or both. The toxin will cause the diarrhea. You know, the toxin will cause the diarrhea. Should I know you the mechanism of action of the toxin? How toxin is causing diarrhea? Toxin have two subunits, subunit A and subunit B. Subunit A, first let me read and then I will draw the diagram. Subunit A facilitates the entry of subunit B after going inside the cell. Subunit B, okay, this toxin have two subunits, A and B. A facilitates the entry of B after going inside. A1 bind with adenyl cyclase and it will split the ATP into cyclic AMP. This cyclic AMP will increase. This cyclic AMP cause more absorption of sodium, which will cause more absorption of water, that will cause diarrhea. Okay, let me draw the diagram for you. So see, this is esophagus, this is stomach. I'm interested in intestine. So this is intestine. Can you see? These are the cells of the intestine. Let me draw few cells. These are the few cells of the intestine. Okay, this is microvilli on them. This is all microvilli on them. This is bacteria. This is our bacteria. This is ETEC. ETEC. So what this will do, this bacteria will produce a toxin. This bacteria will produce a toxin. The name of the toxin is STLT, two type of toxin. This toxin have two fragment, fragment B and fragment A. Fragment B and fragment A. So B for binding. B helps only in binding. B यहाँ पे बैक्टीरिया अंदर नहीं जा रहा है, बैक्टीरिया का टॉक्सिन अंदर जा रहा है. So bacteria ने toxin secrete किया lumen of the intestine. From the lumen the toxin will go inside. B will not go. B will help A to go inside. A after going inside split into two portions, A1 and A2. So inside this enzyme is present. The name of the enzyme is adenyl cyclase. So A1 will activate adenyl cyclase. On activating adenyl cyclase, split ATP into cyclic AMP. So inside this cell, cyclic AMP will increase. All these cells, cyclic AMP will increase. On increasing cyclic AMP, what will happen? What will happen on increasing cyclic AMP? Because of increasing cyclic AMP, sodium is not absorbed inside the cell and water is not absorbed. So sodium and water absorbed nahi hoga, it will remain in the lumen and it will cause diarrhea. Give me a thumbs up. So that is the mechanism of action. Have you got it? Have you got it? That is the thing. So it inhibits reabsorption of sodium and water. Cyclic AMP on increasing the cell. Give me a thumbs up if you got it, everyone. So that is the mechanism of action of ETEC. ETEC diarrhea कैसे कराता है? ETEC. Coming on the third type. The third type EPEC हो गया, ETEC हो गया. EPEC microvilli को damage कर रहा था. माइक्रोविलाई को डैमेज कर रहा था. इट वॉज एडहरिंग, एडहर करके माइक्रोविलाई को डैमेज कर रहा था. ETEC टॉक्सिन प्रोड्यूस कर रहा है. वो खुद नहीं जा रहा है, उसका टॉक्सिन सेल के अंदर जा रहा है. एंड टॉक्सिन आफ्टर गोइंग इनसाइड इट इज इंक्रीजिंग साइक्लिक AMP इन द सेल बाय एक्टिवेटिंग एन एंजाइम नोन एज एडेनाइल साइक्लेज. उसका भी A1 कंपोनेंट, A2 नहीं, B नहीं. हैव यू गॉट इट? कमिंग ऑन द थर्ड वन, EIEC. गिव मी अ थम्स अप. EIEC, एंटेरो इन्वेसिव. इट इज लाइक शिगेला. ये वाला शिगेला के जैसा है. ये वाला शिगेला के जैसा है. So what it will do, it will actually invade inside the cell and destroy the cell and causes diarrhea. Destroy the cell and causes diarrhea. Okay, let me draw the diagram. So it is very easy. This is the intestine. Okay, let me draw few cells in the intestine. This is a cell, this is a cell, this is a cell. Now this is the bacteria. This is the bacteria. It is EIEC. So actually bacteria do not produce toxin, and bacteria do not bind with the microvilli. It is not binding with the microvilli like the first two. The bacteria actually go inside, invading inside, and after going inside, it is destroying the cell. So that the patient will have diarrhea because of invasion. तो ये एक्चुअली खुद बैक्टीरिया इन्वेड कर रहा है. दैट्स व्हाई नोन एज EI, invasive. Have you got it? That is the third. It is like Shigella. It is like Shigella. Have you got it? That is the thing. So EPEC हो गया, ETEC हो गया, EIEC हो गया. Coming on the fourth, EHEC. Give me a thumbs up. EHEC, enterohemorrhagic. Hemorrhagic. So it will again produce a toxin. The name of the toxin is virotoxin. Virotoxin. It will produce a toxin that will cause hemorrhagic colitis. So it will produce dysentery. The blood is also present in the diarrhea. It is also cause HUS. So very important. It will produce a toxin, not ST or LT. It will produce virotoxin, Shiga-like toxin. So what it will do? It will go. The toxin will go inside the intestinal cell and inhibit the ribosomes and inhibit the protein synthesis inside the cell that will cause the diarrhea. Do you want me to make the diagram? Okay. This is esophagus. This is stomach. This is intestine. I am interested in intestine, right? Okay. So let me draw few cells in the intestine. These are the few cells. Let me draw the toxin. This is the toxin. Which one? This one is EHEC. So what EHEC will do? EHEC will not go inside the cell. It is not invasive. It will produce a toxin. The name of the toxin is virotoxin. That virotoxin will go inside the cell. After going inside the cell, it will inactivate ribosomes. So ribosomes are inactivated, so no protein synthesis inside these cells. Cell will be destroyed and it will cause diarrhea. So diarrhea is due to inhibition of protein synthesis inside the cell. Give me a thumbs up. That will cause the diarrhea. That will cause the diarrhea. Done. That will cause the diarrhea, and it is the most common cause of HUS. Give me a thumbs up. उसमें भी एक special strain है जिसपे बार-बार repeated PYQ आता है. You know the answer. It is O157H7. You know two antigens O and H. तो O157 और H7 वाला strain सबसे ज्यादा HUS कराता है. That is the thing. Okay. And last one is EAEC. Enteroaggressive. Enteroaggressive. It is the second most common cause of traveler's diarrhea. Traveler's diarrhea. You know, most common cause of traveler's diarrhea ये है. Second most common ये है. ये children में कराता है. ये travelers में कराता है. ये second most common travelers में कराता है. ये HUS कराता है. So you should learn most common also. Done. And okay, what is the mechanism of action? It also produces a toxin. The name of the toxin is yeast. Enteroaggressive stable toxin. EAST. Done. What does toxin do? What is the mechanism of action of this toxin? It causes diarrhea. The exact mechanism is not written here. It will adhere to the intestinal cells and take brick appearance, and it will cause diarrhea. Done. That is the mechanism of action. Now let me enumerate the 5 mechanism of action and show you the question again. So tell me, enteropathogenic क्या था? It was destroying microvilli and causing the diarrhea. Enterotoxin क्या था? It was producing ST or LT. ST toxin था, ST और LT, and ST and LT after going, the toxin after going inside the cell, it was stimulating an enzyme adenyl cyclase और guanyl cyclase, आप दोनों बोल सकते हो. So it is increasing either cyclic AMP or cyclic GMP, and it is causing diarrhea. Because of cyclic AMP and GMP, sodium water reabsorption do not take place and it will cause diarrhea. The third is invasive. Actually the bacteria will invade. Bacteria खुद अंदर जाएगा, उसका toxin नहीं. Enterohemorrhagic produce a toxin known as virotoxin, like Shiga-like toxin, and enteroaggressive produces a toxin which is known as yeast. तो टॉक्सिन वाले आपके 3 हो गए, एक ये वाला, एक ये वाला, ये वाला. ये आपका टॉक्सिन नहीं बनाएगा, एडहर करेगा. ये आपका टॉक्सिन नहीं बनाएगा, इन्वेड करेगा. सो हैव यू गॉट इट? गिव मी अ थम्स अप. गिव मी अ थम्स अप. गिव मी अ थम्स अप. Everyone give me a thumbs up. First give me a thumbs up, so I will show you the question again. I will show you the question again. Okay, so this is the question for you. Just a second, let me go back. Okay, let me go back. Where is the question? Now you can solve this question very easily, I guess. So here is the question for you. Okay, so this one. Now match the column. Now you do the match the column. Now see which 4 are asked: ETEC, EIEC, EHEC and EPEC. Now read the question. It is heat stable, ST. एंड गुइनाइल साइक्लिस और एडिनाइल साइक्लिस। ये कौन करता है? ये कौन करता है आपका? ये आपका ईटीईसी करता है। तो ए का आंसर हो गया 1, है ना। इन्वेड इंटेस्टाइनल म्यूकोसा, इन्वेड, इन्वेड कौन करता है? इन्वेड ईआईसी, आई फॉर इन्वेजन। तो ये 2 का 2 ही हो गया, राइट। टॉक्सिन अगेंस्ट वास्कुलर एंडोथेलियल सेल्स, यू नो दैट इज वीरोटॉक्सिन आपका, दे आर आस्किंग फॉर वीरोटॉक्सिन। सो ये आपका ईएचईसी हो गया, राइट। और एडहर्स टू द माइक्रोविलाई, ये आपका पैथोजेनिक वाला हो गया। सो ए का 1, बी का 2, सी का 3। सो आंसर वाज इजी। ए का 1 है, बी का 2 है, सी का 3 है, डी का 4 है। उल्टा सीधा भी दे सकते थे वो, तो यू कैन मैच द कॉलम। गिव मी अ थम्स अप एवरीवन नाउ। नाउ एवरीवन गिव मी अ थम्स अप। यू गॉट द क्वेश्चन अगेन? Should I proceed to the next question now? So not only this, any question can come. ये तो चारों का mix and match आ गया. Now the question can come like this. हो सकता है इस बार एग्जाम में ETEC पे क्वेश्चन आए. What is the mechanism of action of ETEC? The 4 options are in front of you. What is the mechanism of action of EIAC? The 4 options are in front of you. So you can pick your answer. Likewise, so there are many potential questions hidden inside this one question. So you should be able to solve all of them. Let me move to the next question. Give me a thumbs up if you got this, everyone. Okay, I hope you got it. So this is the next question for you. This is the next question for you. Read the question and tell me the correct answer. Read the question and tell me the correct answer. What is the correct answer? Okay, read the question. So what is the type? Which of the following type involved in initial response and type 1 hypersensitivity? Now from immunity in micro immunity section, hypersensitivity is a very potential topic in which repeated MCQs are asked, repeated PYQs are asked. So read the options first. So you have 4 options. Write your answer in the chat box. Histamine release, leukotriene and prostaglandin release, mast cell activation or neutrophil activation. Which of them are correct? ABC or ACD or AD or BCD? What is the correct answer? Give your answer from these. These are your options. Options ये हैं, options ये नहीं है. इनमें से select करो क्या क्या सही है. Multiple options are correct. So select your options and tell me what is the correct answer. Tell me, any one of you. I'm asking about type 1 hypersensitivity. You should know the mechanism of type 1 hypersensitivity. Yes, anyone? Anyone? Am I visible? Am I audible? Give me a thumbs up. Write your answer in the chat box. Why you people are not writing? Write your answer in the chat box. I can't see your answer. Write your answer in the chat box. Anyone? What is the correct answer here? What is the correct answer here? Okay, the correct answer here is A. That is A, B, C. These 3 options are correct. I will come on the question again. Let me tell you the mechanism of type 1 hypersensitivity. It will take only 2 minutes. Listen, everyone. The mechanism of action of type 1 hypersensitivity. We know type 1 hypersensitivity includes all allergies. All allergies like asthma, like hay fever, you know, like atopy, like angioneuritic edema, like all allergies are included in type 1 hypersensitivity. So what is the mechanism? I would like to draw a diagram for it. Okay, so this is the antigen. It is entering in human body. Just suppose I am the patient and this is the antigen I am inhaling. The antigen can be a pollen grain, it can be a dust, it can be any particular thing for which this is an allergen for me. So this allergen is I am inhaling from nose. So it is going inside my respiratory tract. So the first cell which come in contact with this antigen is known as dendritic cell. Dendritic cell, also known as antigen presenting cell. Antigen. So the antigen is engulfed by this cell. It will engulf the antigen, degrade the antigen into multiple pieces and process the antigen on the surface with the help of MHC. MHC, you know, major histocompatibility complex pair. It will process it on the surface and it will take this antigen to another cell. The name of the another cell here, the second cell is T cell. T lymphocyte. There are 2 types of lymphocytes, CD4 and CD8. So it is CD4 T lymphocyte, which is helper. TH2 type of helper cell. So exact learn karo. B की T, it is T. T में CD4 की CD8, it is CD4. CD4 में helper cytotoxic की helper, it is helper. Helper भी 2 होते हैं. TH1 की TH2, it is TH2. Question can come on anything. So it is TH2. So this cell will present the antigen to this cell. So it will get stimulated. And on stimulation, it will secrete 3 things from it. Interleukin 4, interleukin 5 and interleukin 6. It will secrete. Out of this, interleukin 5 will go to B lymphocyte. So let me come on the third cell. The third cell is B lymphocyte. It will stimulate. Interleukin 5 will stimulate B lymphocyte. B lymphocyte will get converted into plasma cells. They will get convert into plasma cells and plasma cells will form antibodies. You know, antibodies are formed. There are 5 type of antibodies: IgG, IgA, IgM, IgD, IgE. Gamma. Out of the gamma here IgE is formed. Last one, E. IgE is formed. Gamma में से सिर्फ IgE बनेगी. Now this antibody will go to the last cell. What is the last cell here? The last cell here is mast cell. Can you see here? It is mast cell. On the surface of mast cell, these antibodies will bind like this. Like this. This is FC portion. FC portion से bind हो जाएंगी. So all the antibodies will bind on the surface of the mast cell by FC portion. Now this mast cell is known as sensitized mast cell, right? Now, when the same antigen is entering human body second time, first time to kuch nahi hua, second time. So the same process is not repeated again. Now this antigen will directly go. This time this antigen will directly go to the mast cell. So when this antigen will directly go to the mast cell, it will bind on the surface of the antibodies on FAB portion. So these antibodies will give signal to the mast cell कि antigen aa gaya hai, the mast cell will rupture. This is known as degranulation of mast cell and the granules will come out. Granules contain histamine, serotonin, prostaglandin and leukotriene. All these things, and these things will cause bronchoconstriction and vasodilation that will lead to allergy. That will lead to allergy. So this is the mechanism of action of allergy. This is a diagram. You can see here the same diagram showing, or now this topic is the overlap in patho and micro. You can see the question is from patho and you can see the question is from micro. So it is an overlap question. In both the subjects we have this hypersensitivity, right? So see here, you can see this is the antigen. This antigen is going inside. This is respiratory epithelium. It is coming. This is dendritic cell. Can you see this cell? The first cell it is taking is dendritic cell. It is degrading it, processing on the surface. Then it is taking it to T cell. T cell is helper Th2 cell. It will secrete IL-5 that will stimulate B cell. B cell will convert into plasma cell. Plasma cell will secrete IgE antibodies. IgE antibodies go to mast cell. When same antigen coming again, it will bind to the antibodies on FAB portion. So mast cell will rupture. That is degranulation. Granules are coming out. Have you got it? Give me a thumbs up. So this is the whole mechanism. Many questions are repeated from this. Now I will show you the question. You can tell me the correct answer now. Have you got it? See here everyone. So first time when antigen is coming, it is captured by antigen presenting cell or dendritic cell, which will present it to Th2 cell, which will secrete IL-3, 4, 5. IL-4 causes activation of B cells, B lymphocytes. B lymphocytes will convert into plasma cell. Plasma cell will form antibodies IgE. IgE by FC surface bind to the mast cell. Now these are known as sensitized mast cell. Now when the same antigen coming second time, it will bind to the antibodies, IgE antibodies which are present on the mast cell. So mast cell will damage. It will cause degranulation. From the granules, can you see the granules are coming out? See, when antigen is binding on the antibody, granules are coming out. So because of this degranulation, histamine, serotonin, leukotriene and prostaglandins are coming out. Out of these, you can see, and they will cause the allergy. Have you got it? They will cause bronchoconstriction, increased secretion from the glands, vasoconstriction. They will lead to the symptoms of allergy. Have you got it? Now see the question and tell me the correct answer. Those who were unable to tell me the correct answer previously, now they will be able to tell me the correct answer. Now see the complete, you know the complete mechanism of type 1 hypersensitivity. Now read options, what is correct among them. So is histamine release occur? Yes, it occurs by degranulation of mast cell. Leukotriene release occur? Yes, it occur. Prostaglandin release occur? Yes, it occur. Mast cell activation occur? Yes, it occur. But in the whole process, there is no neutrophil. The first cell is dendritic cell or antigen presenting cell. Second cell is CD4T cell, which is helper Th2 cell. The third cell is B lymphocyte. The fourth cell is plasma cell. And the fifth cell is mast cell. But there is no neutrophil. So neutrophil activation is not there. So ABC is correct. So ABC is given in option 1. So option 1 is correct. Option 1 is correct. You all are right. Option 1 is correct. Give me a thumbs up. Give me a thumbs up. Let me move to the next question of previous year question INICET micro. Okay, so this is the next question in front of you. Read it. It is from virology. It is from virology. So all these viruses can integrate with human genome except, the question contains except, the 4 options are in front of you. Human papilloma virus, HPV, Epstein-Barr virus, EPV, human T cell lymphocyte virus, HTLV, or hepatitis B virus, HBV. So which among the following does not integrate with human genome? So the question is not based on knowledge. The question is based on common sense. If you don't know the answer, still you can answer. Still you can answer. How? You will ask, ma'am, how? We don't know the answer. How we can still answer? Do you know that every one of us know the classification of virus? I guess. Viruses are of 2 types. DNA viruses and RNA viruses. Right? Right. Listen. DNA viruses and RNA. You know what are DNA viruses? What are RNA viruses? Listen. Now this is a human cell. Human cell. Human cell. Not viral cell. Human cell. This is the human nucleus. Inside human nucleus, do we have DNA or RNA? Common sense. Okay, we all have DNA. We don't have RNA inside the nucleus. We have DNA as genetic material inside the nucleus, right? So we all have DNA. Now you tell me which virus, DNA will integrate with human cell or RNA will integrate with human cell? You tell me. Of course it is DNA. DNA से integrate होगा ना. So DNA virus will integrate directly with the DNA of the human. So all of them, all DNA viruses will integrate with human cell, but RNA viruses require an enzyme that is retrovirus or reverse transcriptase enzyme. Before that, RNA is converted into DNA so that it can integrate with the human genome. RNA वायरस डायरेक्टली ह्यूमन जीनोम से इंटीग्रेट नहीं कर सकता क्योंकि RNA वायरस में RNA है. So DNA cannot integrate with RNA directly. Before that, RNA should convert into DNA. आपको RNA को DNA में कन्वर्ट करना पड़ेगा with the help of an enzyme known as reverse transcriptase. So once reverse transcriptase converted, then DNA can combine with DNA, and DNA can directly combine with the DNA. So which of the following does not, except in a question may integrate with human genome? So in short, which of the following is the RNA virus? Which of the following is the RNA virus? The one which is the RNA virus that will not integrate, and all others are DNA that will integrate. So the question is basically based on the classification of the viruses, not with the knowledge. Have you got it? So it is a common sense. Now you know the classification. I guess you all know the classification. Yes, what is the correct answer? No, Madhavan. The correct answer is not 1. The correct answer here is C, because C is a RNA virus. Rest all are DNA viruses. Let me tell you the classification. Let me tell you the classification of the. Now there are 6 viruses which causes cancer in humans. So there are 4 DNA viruses and 2 RNA viruses. 4 plus 2. Total 6 viruses causes cancer in human. These are known as carcinogenic virus, right? So what are DNA viruses? Among DNA, it is HBV, that is hepatitis B virus. HHV8, human herpes virus 8, it is also known as Kaposi virus, Kaposi sarcoma virus. HPV, human papilloma virus, and Epstein-Barr virus. These are the 4 DNA viruses. C, RNA are only 2. You have to learn the names, HTLV and hepatitis C. Hepatitis 2 हैं, hepatitis B और C. C आपका RNA है और B आपका DNA है. These all can integrate directly with the human genome. But these two require a reverse transcriptase enzyme for integrating. They do not directly integrate. Have you got it? Have you got it? To cause the cancer. Before integrating, without integrating, they cannot cause the cancer. Now see, you can see the options here. So out of this, HPV, EBV, and hepatitis B. These 3 options are given here, no? You can see here. So it is HPV given in your option, HPV and EBV. These 3 options, but 4th option is HTLV, which is the RNA virus. So our answer is HTLV. Give me a thumbs up, those who were wrong specially. Give me a thumbs up, especially those who are wrong. Give me a thumbs up, you got it. Have you got it? Yes or no? Yes or no? So that is the correct answer. So correct answer here is C. So this is the mechanism for DNA and RNA viruses, how they are oncogenic. So for DNA viruses, they directly integrate. Can you see? This is a human cell. Everyone see. This is a human cell and this is the nucleus, and blue color is the human DNA. And this is the virus, DNA virus, which is entering inside the cell. The red color is the virus DNA. So red and blue directly combine with each other. That is, integration takes place directly because both of them are DNA. Virus may be DNA, red color DNA. And human may be DNA and the blue color DNA. And both of them are integrating with each other, causing mutations, causing cancer. This is how DNA virus is causing cancer. And among these are the 4 DNA viruses which causes cancer. And among RNA viruses, they require an enzyme known as reverse transcriptase. See the enzyme. So see, this is a human cell. Inside the human cell, see the nucleus. The blue color is the DNA. DNA of human. And this red color is the RNA. RNA of the virus. Now directly RNA cannot integrate with the DNA, right? So it requires an enzyme. Can you see the green color enzyme? The name of the enzyme is reverse transcriptase. So reverse transcriptase first convert RNA into DNA. It converts RNA into DNA. So such virus in which RNA is converted into DNA, this is known as provirus. Now it can integrate and and cause the cancer. This is how we can go with it. Have you got it? Next question. Next question in front of you. Please read. Everyone read this question and tell me what is the correct answer. Who will tell me what is the correct answer? It is a very easy question. Please read the question and tell me what is the correct answer. Which of the following is associated with aplastic crisis? Again, the question is based on previous year virology. So parvovirus, Epstein-Barr virus, adenovirus, or hepatitis B virus. Which of the following virus is associated with aplastic crisis? Yes, aplastic crisis or aplastic anemia. What is the correct answer? I'm giving you 10 seconds. Please write your answer in the chat box. The 4 options are in front of you. Parvovirus, Epstein-Barr virus, adenovirus, or hepatitis B virus. Yes, all of you are right. The correct answer among them is parvovirus. The correct answer among them is A, that is parvovirus. What are DNA viruses? There are only 6 viruses which are DNA. 6 viruses. All other are RNA. You should know the name of DNA viruses. So it is double H A triple P. Double H A triple P. What are 2H? Hepadina and herpes. What is 1A? Adeno. What are 3P? Parvo, Papilloma, pox. So these are the 6 viruses which are DNA. Remaining all viruses are RNA. So you should learn the classification first, right? Among them, parvovirus. Let me teach you parvovirus. Parvovirus is a DNA virus. All these DNA viruses double-stranded DNA virus except parvo, which have single-stranded DNA. So again, important MCQ or PYQ. It is a single. It is the smallest virus, smallest DNA virus, smallest overall virus. See, it is the smallest virus. Again, a PYQ. Parvo पे क्या क्या क्वेश्चन आते हैं? It is a DNA or RNA? Of course, it is a DNA virus. It is single-stranded or double-stranded? It is single-stranded DNA virus. It is smallest virus, smallest DNA virus also, smallest overall virus also. Let me tell you, parvovirus is a family. इसमें एक ही virus आता है, parvovirus B19. This family have only one important virus, that is parvovirus B19. Transmission कैसे होता है? Either by respiratory route or by vertical route. Vertical route is from mother to fetus via placenta, right? What is the pathogenesis? Listen, I will tell you the pathogenesis. This is a bone marrow. This is a bone marrow. Bone marrow contains hematopoietic stem cell from which all RBC, WBC and platelets are formed. It is a precursor of all cells. From this hematopoietic stem cell, various stem cells are formed. So this is a stem cell for RBC from which ultimately RBC is formed. This is a stem cell for WBC and this is a stem cell for platelet, right? So when parvovirus enters in human body, this is parvovirus. So this parvovirus go to the bone marrow and inside the bone marrow it binds to the this parvovirus bind to the RBC stem cells. So RBCs are not formed. WBCs are formed, platelets are formed, but only RBCs are not formed. So patient presents with aplastic anemia. So this is known as anemia. Anemia. So patient have only anemia, no leukopenia, no thrombocytopenia. Patient have only anemia, but that is due to that the stem cells are not functional. That is known as aplastic anemia. This is known as aplastic crisis. Have you got it? Have you got it? So B19 virus bind with the P antigen on the erythroid precursor of the bone marrow. Erythroid यानी RBC के precursor. So it causes cell lysis of the erythroid stem cells. So it will causes anemia, that is known as aplastic crisis. Give me a thumbs up. So I will tell you 3 diseases. The name of the 3 diseases. Name the disease caused by parvovirus in children. Name the disease caused by parvovirus in fetus. And name the disease caused by parvovirus in adults. Can you name them? 3 diseases. 3 questions, 3 answers. Anyone? So in children, it causes 5th disease, also known as slapchick. You know, gaal pe chanta mara. I will show you. In fetus, it causes aplastic crisis. In adults, it causes arthralgia. Okay, I will show you all. So let me talk about children first. In children, it causes 5th disease, also known as erythema infectiosum, also known as slapped cheek. ऐसा लगता है गाल पे किसी ने चांटा मारा. Can you see? Slapped cheek. So slapped cheek disease. It is that in children. Have you got it? In adults, it causes arthralgia, especially wrist joint. Wrist joint. There's a PYQ in your NEET PG. Done. So you have to learn. In fetus, it causes aplastic crisis. Aplastic crisis, fetus will die because of severe anemia known as hydrops fetalis and abortion will occur. So that is the thing. That is the thing. Have you got it? Have you got it? So I am done with this. It is only episode 1 in which I have covered nearly 12 MCQs now. Still we are remaining further more than 20-30 MCQs are having with me. I'm covering both papers, right? I'm covering November 2020 also and I'm covering June 2021 also. So I'm having many more questions. So I will take episode 2 of it on Monday. So don't forget to join me Monday morning at 10 a.m. not 10:30, at 10 a.m. in the morning. There will be a lecture, that is episode 2 of this Microbiology INICET PYQs. So thank you very much for being with me. If you like the lecture, don't forget to click on the like button. Please drop a comment in the comment box. Your feedback, your suggestion are really important for me. If you have any suggestion, any feedback, please write it in the comment box. I can see your comments. And don't forget to share with your friends. Thank you very much. And don't forget to subscribe our YouTube channel. Let's crack NEET PG. Thank you. All the best. See you all on